Yep.
Hi, everyone. Thank you for joining us today. We can start off with a round of introductions since we have new people here. So we can maybe do name, where we are right now today, and one fun thing that you are up to. So I can start. My name is Aish. I am right now in India, and one fun thing that I'm up to is trying to learn Arabic. And I'll choose the next person. So you can choose the next person who's on your screen or whoever's not gone. So I choose um, uh, Adriana.
Hi, I'm Adriana Bernal, and I am from Colombia. I work at Universidad de los Andes. This is my first time in this meeting. And we are interested in trying to become more independent from the commercial enzymes and kits that we get, because we always get them. It takes a long time to get them, and also we usually get less efficient enzymes in the by our providers in Colombia. And what am I up to? I don't know what to say. I think that's it. Thank you.
Adrian, would you like to nominate someone to introduce themselves?
Sorry. Okay. Deborah.
Up to me. Okay. Hi, I am Deborah Oluwasonia. I am currently in the United Kingdom. I am helping to coordinate the Reclone project. One fun thing I would say I am up to these days is I am learning a new computer language. Yeah, as if I don't have enough on my plate already. So I decided to take up the challenge. I want to learn how to use Perl and also a little bit of MySQL. So I will nominate Guy.
Hello, I'm Guy Edelberg. I'm in Paris. And I just finished a PhD on democratizing DNA detection, nucleic acid detection, and LAMP stuff. I've also been making some of these enzymes. And I'm right now reading, there's a new Ellington paper, how they even further improved the BST with AI and made a bunch of mutations. And I have students in the lab that are trying to detect a specific fungus that's attacking a lot of trees in France now. Things like that. And then I'll nominate Fernan.
Hi, I'm Fernan. I work in Chile. We are working with a number of people collectively in the Jogel Open COVID initiative and the Open Bioeconomy Lab also to create our own homebrew LAMP reaction, and also in some projects for working for RT-PCR reactions, following the same limitations that Adriana was describing, basically autonomy in the access to enzymes and good enzymes that we know how they perform. And I nominate Robson.
Hi everyone, my name is Robson Tramontina. I'm talking from Brazil, and I'm a PhD student from the University of Campina, and also I'm the founder of Equa Biotech. I'm mostly interested in protein production, especially for diagnosis purpose and molecular biology tools. I've been following this meeting and this community for a quite long time, but doing a lot of work I've been doing, I have never the opportunity to be more talkative here. So I'm really glad to be invited to give a lecture today. And I hope you guys can understand a little bit what I'm doing here. And I'm quite excited to get a feedback from you guys. So I will nominate Janie Molly.
Hi everybody, good to see you all. I'm Jenny, I'm based in Cambridge in the UK, and I have a group at the University of Cambridge who are working on local manufacturing of enzymes. I also work very closely with Stefan and the work that's going on in Maboa Lab as part of a Beneficial Bio, which is a network we're trying to build of small businesses that are setting up to manufacture and also distribute reagents to try and overcome some of the supply chain challenges that we all know about. So really excited to hear the talks today, and yeah, great to see some new faces. Thanks so much for coming. Forgot to nominate someone. I was 2 minutes late, so I'm going to say Natalia and hope that she wasn't the first person and I missed her.
Hi, hello everyone, I'm Natalia. I'm based in Colombia. I work for a university in biotechnology and biomedical engineering programs. And I joined this network since I assist to the BioSummit, which is an initiative that wants to democratize biotechnology and empower local educators. So I met most of these people there, and especially regarding COVID, I've been working somehow in identification in wastewater of the COVID as an early warning system. And I've been also working in some epidemiology genomics, analyzing some DNA sequences that like a group of universities are generated. And I'm also trying to join people to work in the local manufacturing of recombinant proteins for diagnostics. And sorry, I also was be a little bit late. I don't know who please can help me to introduce himself if have no time to do it yet.
Okay, I can jump in to help. Maybe David Duplat.
Thank you.
Well, hi everyone. I'm David Duplat from Colombia as well. I'm a biologist and a microbiologist from Universidad de los Andes, just like Adriana. Well, currently we are working in a project involving the University of Toronto, and we are working with Fernan as well. Actually, he's the one that referred me to these seminars. Really don't have a lot of experience in this topic, since this is all new to me, but I'm really excited to learn this stuff, as it's interesting for me and important for our country, as Adriana previously mentioned. And yeah, well, I'm happy to be here. And I think everyone has been nominated. I'm not sure if anyone is missing.
There is Hortense, there is Stefan, and there is Brian Mutubu.
Okay, so I will nominate Brian.
Okay, I can go on. Hello everyone, this is Brian Mato from Makerere University. Currently I'm completing my master's in immunology and clinical microbiology. I really have a biomedical engineering background with key interest in diagnostics, and to see really how best we can have a lot of innovative products from Africa, and basically they used in Africa and made from Africa. I'm really here. I think this is my second meeting to be part of Reclon, and for today I'm really excited to learn about from experience, from other people's experience, to see to pick something and see how best probably we can have it translated or to Uganda and maybe a few other areas, and then also to really create network. Thank you. I can nominate, I don't know, Guy Paris, hope I'm right.
Maybe I can help. The options is David, sorry, no, not David, Stefan, and I think I saw Hortense, but I think Hortense is gone off. So maybe I think we're left with Stefan. Is that right, Aish? Yeah, okay, Stefan. Okay.
I think Stefan also just left. So
he should be coming back because he's speaking. So
yeah.
I think probably the internet just went.
Yeah, that is right. And anyway, I can jump in to help. Hortense, welcome back. Would you like to introduce yourself? What we're doing is we're introducing ourselves, saying where we are at. Maybe I'll let Aish go take it up from here.
Yeah, so where are you, what's your name, where are you from, and what work are you up to, or something interesting that you're up to? We can have Stefan, if you're back, or Hortense. Okay. Harry.
Hey, I don't know if you can hear me.
Yes, we can.
Okay, I'm Hortense. I'm working with Guy Heidelberg at the CRI in Paris. I'm working as an engineer and I try to help him as much as possible. And I'm from Paris.
You are welcome. Harry, would you like to introduce yourself? Let us know if you're there. Let us know where you are. And one fun thing you've been up to, if you're able to.
Sorry, is this supposed to be Harry to introduce himself?
Yes.
Sorry, just rounding up some stuff in the lab. So my name is Harry. I'm based in Ghana, part of the Open Bioeconomy Lab nodes in Ghana called Hive Biolab. We are into the local biomanufacturing of enzymes, particularly those found within the Open Enzyme Collection. And currently making use of the Open Enzyme Collection to as a way of locally producing some of the enzymes in there and testing them as diagnostic polymerases for identifying some sexually transmitted infections in the multiplex assay that I have developed. And I'm doing this as part of a master's thesis at the Kwame Nkrumah University of Science and Technology. I'm currently based out of Kumasi in Ghana, where we largely are leveraging the Hive Biolab to solve community problems using biology. So that is what I do, and that's why I have other colleagues currently in the lab, but they are currently performing some experiments. So unfortunately, they will not be able to say hi, but I'm here, so great.
Great. So thank you everyone for coming. Thank you for these fun things, fun projects that everyone is involved in. So today, which is the day we've been expecting, we are having two exciting talks. We're going to be learning about what currently ongoing in the Global South to encourage or to make reagents more accessible. So we're going to have Stefan Vandanka. Hope I called it correctly. He is the manager of Beneficial Bio in Cameroon. And after that, we're going to have Robson Tramutina, if I called it correctly. He's also going to be sharing a bit about his journey in Acrobiotech. So before I start, I'm just going to read a little bit about Beneficial Bio and introduce Stefan. So Beneficial Bio is a network of social enterprises run by biologists with a goal to help labs around the world secure reagents quickly and economically. The Beneficial Bio node in Cameroon is based in the Mboa Lab. Mboa Lab Biotech is based in the Mboa Lab. So the biotech enzymes and reagents are locally produced to provide researchers with tools and services quickly at a fair price and with specialized local support they need to do their best science. I could put the link to the Beneficial Bio website in the chat after I do the introduction. Now to our speaker, Stefan. Stefan Fadanka is the manager of Beneficial Bio at the Mboa Lab in Yaoundé, Cameroon. With a background in molecular biology, that's an MSc, and biological analysis as his bachelor's. His current research interests includes the screening of novel, cheap and environmentally friendly biologically active compounds from microorganisms, and the development of cost-effective and widely accessible plant propagation method. He is a member of the Open Bioeconomy Lab project. So with that, I would hand over the virtual podium to Stefan for his talk. And after that, we're going to have a short question and answer session. So over to you, Stefan, and you're welcome.
Thank you. So I'm just trying to share the screen. I hope everyone can see the screen.
Yes, we can.
Okay. Thank you very much. Hi, everyone. Thank you very much, Deborah, for the introduction. Yes, so my name is Stefan Fadanka. I'm the Executive Director of the Mboa Lab based in Cameroon, also Research Manager at Beneficial Bio. Yes, I'm very happy to be here today and share what we've been up to and our experience in local manufacturing of enzyme in Cameroon. So I will just go ahead with the presentation. Okay. 1 minute. Okay. So I will just briefly introduce the Mboa Lab. So the Mboa Lab is a Cameroon-based community innovation hub. The lab was founded in 2017 by Dr. Thomas Mboa. And really the aim of the Mboa Lab, the aim and mission is actually to catalyze local sustainable development through open science. And aside from the aim, Mboa Lab also has a very strong vision, which is we advocate for the local sustainable development. That's a development really tied or bound with the fact that local people that need or people are at the origin of that development. So it's something that is very important as the vision of the Mboa Lab. So it's important to include people in order for the development to be sustainable. And we do that through various activities such as workshop and capacity building. So we organize mostly workshops. So we work as a community innovation hub. We organize workshop with people from various age groups and even background. I also do capacity building for students and local researchers through internships and yes, placement and workshop as well. We also do a lot of open hardware design and prototyping. So maybe through the presentation, you will see some design that were locally developed as well, because amongst the limitations in terms of resources, equipment takes or have a very big place as well, aside from the reagents. And then we also do a lot of research projects. We are involved in many research projects as part of the activity we perform on a daily basis. So currently we are involved in the UK Global Challenge Research Fund projects to develop the Cas12 typhoid DX. Basically, it's a molecular diagnostic technique based on the Cas12 technology to diagnose Salmonella or the typhoid fever. We also have some other projects funded by the Open Society Foundation, basically to develop affordable, low-cost medical hardware devices. And also more recently, we are involved in Volkswagen Stiftung, projects to locally manufacture reagents and kits for the detection of SARS-CoV-2 virus. And also we are involved in local enzyme manufacturing. So I think this presentation will be mostly focused around that, our activity toward enzyme biomanufacturing here in Cameroon. Next slide. Okay, so I just go ahead and to answer to the question why local manufacturing of reagents. So being of biology, biotechnology background, and then we really lived some challenges while performing our research and doing our work as maybe master student or at the university. And we realized maybe later on that these challenges were faced by many other people around us. And there were some recurrent context-specific issues faced by researchers around us in Cameroon. And then it's like I mentioned, it's later on that we saw that these issues were not that context specific because many people around the world are suffering or are challenged by the same issues. So these are the costs, mostly the cost of reagents, mostly for molecular biology reagents. Most of them are quite costly and it's difficult to have them in local sourcing for local procurement. Also the delivery time. So even when you manage to actually buy the reagent that will maybe be from countries in the West and aside from Africa, you have to wait for at least 3 months. From personal experience, for example, I had to wait close to 2 to 3 months to have the reagent I've ordered. So it's a real issue. Also lack of some specialized services. Maybe some recent or specialized services such as sequencing, for example, are very limited. So we have very limited facility for that. So most often we have to like look outside and send a sample to be sequenced. The overall problem is creating a lot of delays. And many people are even often obliged to drop out. Some projects are obliged to stop because of the delays and maybe the cost of some equipment or reagent mostly. And overall, it limits greatly the research that can be done, even the participation to maybe Africa, Cameroon in research, more specifically in molecular biology. So overall, we were trying to, we identified that there is a real reagent access problem. And then it's not only limited to reagent, but equipment as well, as we will see as the presentation will unfold. I think it's very well summarized in an article published in 2029, a blog post from Lara Strip. So it's called Enzyme Toolkit Made at Stanford Health Molecular Biotechnology Globally Accessible. So it really explained the situation, what we face here. Also how the open enzyme collection that was developed by the Open Bioeconomy Lab in collaboration, as the article would say, in collaboration with the University of Stanford and some other research groups, to come up with the open enzyme collections. And as part of the Open Bioeconomy Lab, we were very privileged to be using the open enzyme collection at a very early stage. And doing that, we pioneer local enzyme production in Cameroon. So we've been able to actually use the collection to express and produce the reagent. We started by DNA polymerases that we used. And so we've been able to do that. And basically we solved the issues we've been facing. And at one point, the question was like, what next? So we started thinking about the bigger pictures. How do we harness the potential that we already had to do something bigger and create more impact? So, and then I think the solution really came with Beneficial Bio. And since we had already some facilities with to do local enzyme production, so in 2019, the Mboa Lab became a biomanufacturing node for Beneficial Bio. So Beneficial Bio is a network of social enterprises run by biologists, and then our goal actually is to help laboratory researchers in the world to secure reagent quickly and economically. So we insist on the two because the advantage of local production mostly it will help to speed up the production, reduce the production time, the delivery time, and then some even the cost of the reagents. So that's what the challenges we are mostly targeting, but we also do a lot of other things, as you will see. Basically, we provide researchers, scientists in Africa, Latin America, and elsewhere in the world with tools and services quickly at a fair price and with the specialized local supports they need. Because the reagent access problem is like a part of a bigger problem, because in our context, research is also limited by the lack of some services, as I talked earlier, but also the support. You might have access to a reagent, but not necessarily all the technical support around that to be able to best use it. So yes, so at Beneficial Bio, what we do local production of enzyme and reagent to speed up quick delivery time. Normally, we can deliver reagent in 3 days. And so it has really cut down the delivery time. And we are mostly working towards reagent that can be cold chain free, stable at room temperatures. Also, we do a lot of customer support. So, and also capacity building, as I mentioned. We have on the picture, we can see JC is our intern from the Catholic University, a local university. So we do a lot of trainings, workshops. We take interns to train them in the advanced molecular biology and local production techniques. And also for customer support, we develop a series of tailor-made and maybe context-specific methods to be able to interact the best way with the customers. And also, we do a lot of research and development to create more and more new product that will meet the need for the customers. So yes, here we can mostly see a bit of timeline about Beneficial Bio. In 2019, really, BenBio was established. So we are already going to the third year of active work towards local enzyme manufacturing and creating, helping researchers here to do their best science. So we started by market research to identify the needs, a bit of the needs, where to target, and the importance of doing what we do. Also, we started by the lab capability setup. So we had to do so to implement a local a research and biomanufacturing unit based at the Mboa Lab in Cameroon. And then we went ahead with supply chain setup. So it was a step-by-step procedures. It took time, but yes, slowly but surely we've been able to put a complete biomanufacturing unit with research and development, QA, QC, sales and marketing department. So, and then we did also a lot of social media awareness, and we have some numbers. So maybe we are close to 5,000 followers on Twitter, 23,000 on Facebook. So there's also an extensive presence on social media. And we set up an e-commerce. We have a website where you can have access to the catalog, the products that we are available on our hand. And then we also have some sales locally. So we have procurement to already delivered reagent, or we worked with some corporate clients. We also have some orders from students, some private and public research laboratories already. So the sales are online. And the good news is that in 2020, the Beneficial Bio was ISO 9001 certified. And yes, as you see in the next slide, now we are currently leveraging cutting-edge technology towards the cell-free protein expression to create more product and maybe speed up again our production. Next slide. So I would like to talk a bit about some achievement since the establishment of Beneficial Bio in Cameroon. As I mentioned earlier, we've been able to set up a complete biomanufacturing and research unit with almost all the departments that are needed, from QA, QC, R&D to sales and marketing here in Cameroon. And yes, we've been able to make available to produce some local reagents. So we focus on DNA polymerases. We are mostly working now on research on enzyme, PCR master mixes are already there, DNA loading dye and electrophoresis buffers. And we also, it's important to mention here that in 2020, most of the work was done on through cell-based protein expression. And then now we are working mostly to leverage the cutting-edge innovation in cell-free protein synthesis technology and other manufacturing innovation as well. So using as fermenters and even solid surface protein expression, and that will make we greatly improve what we do. And as I mentioned again, we are focused on, we make sure that the reagents are stable at room temperature because the cold chain maintenance is a real problem here for us. And we did not limit ourselves to reagent because as I mentioned, we started by trying to solve our own issues. And among the issues, we also had a lot of issues with hardware. So we, based on the setting we have a like complete electromechanic department here in Boa Lab. So we've been doing some prototyping of equipment as well. So we also investigated some local hardware manufacturing. And then we started by the tools and the equipment we mostly need for the protein expression. So we prototype, as you can see here, a shaker incubator, and that we use already in the lab, and the desiccator. So we started by the equipment that are mostly used, that we use in the lab. And the idea is to maybe finalize, go to the maybe engineering validation, through engineering validation model, to have robust equipment that can be maybe accessible to researchers in form of a toolkit for protein expression, for example. And also some achievement that is quite important to mention here for us in our context. As you will see in some of the challenges, we have a big issue through awareness and trust from the local awareness from government, big government institutions, and even from customer locally. So we took a lot of time to like develop a strong local presence, most specifically in Cameroon. We developed a technical support and assistance, like we mentioned. We make sure we interact a lot with the customers. And to do that, we came up with maybe interaction through WhatsApp, social media, which is a kind of new. Also, flexible payment method, because our customers are mostly from whiteboard. We have many people from corporate client to the student. So we came up with a mobile money payment method, so people can now pay with their mobile phone, and it's a bit easier for them to have access to the reagent they need. And it's also speeding up some of the processes. but it's also okay to have access to the reagent by through online payment method or Visa payment to credit card. Also local collaboration, which is also one of the bottleneck for maybe startup in Africa. We try to build a lot of local collaboration with local university, be it public and private. So we take students, we interact a lot in research projects with universities, even state research institutions. So we currently have a number of projects done in collaboration with some governmental institutions in Cameroon, and it took time to build that as well. So I'll just move to the next slide. So talking next, we also have some challenges. Amongst them, we have some lack of basic resources. So as a manufacturing hub or unit, we need some reagent. We need reagent to be able to transform and produce the reagent we want to produce, but it's difficult to locally source for the reagent. So most often we get the reagent from the West, and we have a lot of issue at the custom to find a carrier that will deliver the reagent on time. And then the situation has been worsened with the current crisis situation with COVID-19. So it's a real problem, a real limitation, the access to the reagent we need to be able to do the local manufacturing. And also, since we are also involved with hardware design, we also have some limitation in having the parts for the equipment we build. So it's quite difficult to have them around. And also, it's true that we have some ways around. We have a backup system for the electricity, but generally in our context, it's very difficult to have stable electricity and internet aspect. So I know it can seem very I don't know, trivial here, but it can be really challenging when you are involved in such work. And another big issue is funding. So currently we lack, or there are no very well established local fund strategy for startup, more specifically startup like us doing with local enzyme biomanufacturing. So most of the funds are coming from maybe external sources. We've been lucky to have support from the Shuttleworth Foundation, the Open Way Economy Lab, and so other organizations, but funding stays and remain one of the big limitations. And without access to local funds through administrative means, and it's quite difficult. So it kind of push us to work more on sales and yes, to be more active toward the sales directions. Another challenge that we would like to mention here is the lack of local awareness and support. So this is mostly support from institutions like at the governmental level, because since we deal with mostly GMOs, genetically modified organisms, in our work, and so we have to be sure and to be to be in line with all the regulatory boards and regulation around that. But in Cameroon, for example, it's very difficult to have well-established stairs and steps to follow. So that support is actually limiting our work. As I mentioned earlier, the custom is quite difficult to deal with custom here, because when you have the chance to buy the reagent from abroad, when it comes, you have to pay maybe 3 times, 4 times the price. And also maybe I would say gaining, that's a challenge, a limitation that has to do with any startup. You have to gain the trust to your market, and we make sure we do that through our QA, QC checks, interaction with customer. We make sure we get a lot of feedback from them, and always and constant improvement in the products and enzyme we produce. And also another challenge is limited local collaboration. as I mentioned here, it's difficult to evolve in this field alone if you don't have collaborators. Once again, we've been very lucky to be part of the Open Bioeconomy Lab group, to be part of many other organizations abroad. But on the local, at the local level, it's very difficult to have that collaboration. There are some institutions maybe locally that are involved in research and others activities that can be related that we can collaborate, but to initiate that contact, to initiate the collaboration is quite difficult. And we've been also working towards that, as you saw previously, with interaction with public and private institutions in the country. So I think on our side, these are the main challenges one can face while dealing with local productions and manufacturing of enzyme. Okay, a brief note about how Reclone can maybe help us toward achieving and improving our work. So I think Reclone, first of all, is a very great exposition platform. So it can give the opportunity to startup like us to discuss and explain, to come and share their experience with others. It can be inspiring. It can also promote networking that will foster research and specific activities. So Reclone can also be a means to foster sharing of information. And then from our participation in Reclone, we've been very amazed by the kind of sensitive and very useful information that we got through the Reclone forum, for example, about protocols. It can be information about all sorts and very useful information that can be used to like speed up your research, because you can have access to maybe a problem and then you not go for it. You can have a direct interaction with specialists from a particular field. So it's it's a very interesting tool. And then maybe why not like talking in the future, projecting us a bit further, why not having Reclone as a central organization, for example, to interact with what I call here corporate, sorry, there's a typo, corporate groups to ease up procedures and service facilitation. Why not? So maybe we can serve as Reclone can serve as a platform to go to, for example, GHA and say, hey, we have this group, and then what can we do to speed up and ease up certain procedures? Because as I mentioned, procurement of reagent is quite challenging, local sources. So if we can have some mechanism in place through whatever means to interact with these big institutions to ease up, to speed up a bit certain process, I think it can be a very good start. Yes, so I will just stop here for now, and yes, happy to answer to any questions, interrogation. Thank you.
Thank you so much. Thank you so much, Stefan, for that wonderful presentation. I think, and to manage our time effectively, we would go straight to the second speaker. Please, if you have questions, please note them. We will come back to the question after the second speaker. And I will quickly introduce Robson Tramontina. So Robson Tramontina is from the ECRA Biotech startup, and ECRA Biotech is a Brazilian biotechnology company with an emphasis on research and development on the enzymology and local manufacture of high quality molecular biology products at affordable prices and prompt delivery for universities and molecular diagnostic laboratory. I will paste the website of ECRA Biotech after I tell you more about Robson. Robson Tramontina has an MSc, and he is the Director of Innovation and Operations at ECRA Biotech. He's a pharmacist, enzymologist, and bioentrepreneur with 10 years of professional experience. Robson has worked in 6 research institutions in Brazil, Holland, Argentina, and the United Kingdom. He's currently a PhD student in science and technology at the at Unicamp. Yeah, at
Perfect.
Unicamp in Brazil. So welcome, Robson, and over to you.
Well, thank you very much for this introduction. And I would like also to thank Stefan for the previous talk. It was really inspiring. And I must say that we have the same problems here in Brazil. And I'm gonna try to be shorter because basically your pain, it's ours here, you know. So I would like to talk today about Ecra Biotech, which is a Brazilian startup developed by me and some colleagues. Basically, this startup initiated its activities after we started a course at the university doing my PhD, which is related to scientific entrepreneurship. And I came out with this idea because here in Brazil, we also have problems with delivery of molecular biology products, and they always come with some problem related to cold chain, longer delivery time, really high prices, especially by the dollar price here in Brazil, and the real, the local coin here, the local money, it's subvalorized. So we really struggle buying those top types of issues, top type of products. So after we finished the discipline, the course, we came out with a lot of support and initiatives. So me and a colleague of mine, I'm going to present later, we came out with more business partners here. And then Ecra Biotech was born with the goal to develop local science, cost-effective and globally minded. We believe that we are able to develop molecular biology technologies at a fair price, but also with the highest level of quality, not like the simplest and cost effective, but also the maximum quality. It's the main goal of Ecra Biotech. And of course, we've been struggling a lot, but after the pandemic started here in Brazil and started to spread, we got a lot of attention and national attention here in Brazil. We appeared on local TV, on national TV, and many initiatives gave us support to actually build a lab. So we are a private lab here. We are independent from any type of governmental or another type of multinational. We are just indie biotech company, and we were able to build this infrastructure to train some people, some staff. We are passing through many modifications, and we are growing exponentially the last years. And I would like to share some of our progress so far. So the strategic management is composed by Emad, which is the guy related to the corporative affairs and the financial and all the bureaucracy related to sales part of the company. My colleague Fabio, which is responsible for he's the director of quality and also the new business development, and me, which is the guy that let's say try to figure out how to produce those type of enzymes, study a lot to develop the best protocol so far. And after we establish the best method, then we start to produce those enzymes. And I'm also responsible to monitor the production. We also have employees working at the company. We have Marcelo, which is our manager. He has a PhD in protein biochemistry, and he's responsible to take care of the lab when we are not able to do that. We have an intern, Viviane, and we also have a technician, Jerusa, which is responsible for the quality control and guarantee of quality here at the company. So what we do here at the Ecra Biotech, we perform basically one of our activity is research and development. So we are capable to promote services related to life sciences in general. We are interested in applying our knowledge to develop specific diagnosis kits enzymology assays, development of enzymology assays, production of recombinant proteins, lots of consulting and bioinformatics studies as well. We perform those type of services because this is let's say, a type of service that give us money to support, even though we don't have sales in that month, but also because by accepting those types of services, we are learning a lot to incorporate those knowledge in our company. So it is really important for us to be a R&D company besides a manufacturer. So what I'm here to talk today is more related to the manufacture of our products, because we produce diagnosis kits and enzymes and RNA and DNA extraction kits. For example, here, as an example, we have one of our products. We produce this is the, one of them. It can be for any of enzymes, and this is another one which we deliver our extraction kits. So we are still producing lots of things. You can check us at the website. You just have to translate this from Portuguese to your language, because we are actually now focused on the Brazilian market. But we just finished our website, and we have like our portfolio over there, and we focus on production of many enzymes. After we are in this actual laboratory, there is around 1 year, so we've been working a lot so far. We developed 14 products during that time. Our first products were one or two enzymes in the first years, and after the COVID-19 advent, we started to produce more because we have more knowledge and we have more time to focus on that. So basically, we produce enzymes which are comprised such as DNA polymerases, RNA transcriptases. We produce enzymes for LAMP reactions. We produce positive control for LAMP reactions, RNase inhibitors, UNG glycosylases. Those products are already all established and have a quality control protocol for development, and they are working really well, and they are already being commercialized. But we also have, we want to go beyond like pure and only single enzymes. We wanted to develop master mixes. Here we call our master mix as Tupã, which is an indigenous word, which the letters means basically that is a molecular biology master mix for molecular detection of diseases. And we wanted to develop master mixes for LAMP reactions, for RT-PCR reactions. We are also working with CRISPR tools and Sherlock detection tools. We are developing some solutions for that. We wanted to, in the future, also prepare molecular panels for respiratory viruses detections and library prep for DNA sequencing, RNA sequencing as well. So I don't know if you guys can check on the website, but there is a barcode to go straight to the products as well. Some of our partners here, we still don't have big sales right now. We have lots of forecasts for doing that in the second semester. But we serve public market and private market, multinational companies, small companies, local companies, universities. So we still have a good clientship here in Brazil to work with. This is basically what we do here. I don't want to get in details, but we can chat later. This is flow chart, what you're doing here, for example, producing proteins. You guys already know how to do that, right? So we have this protocol for the production of any protein, and the formulation, it's gonna change depending on which protein is that. The difference here is that we are performing good manufacturer practice. We also implementing ISO 13485, which is related with IVD diagnosis. So basically, it's more challenging than just performing like a basic research. We have to take a really serious, rigorous control of each step. And we also have to ensure that the lab is working like very, very in a good manner. So this is quite challenging to do so, but we're still learning how to do that. So the basic difference of like producing an enzyme in a research lab in the uni is we have to think like a manufacturer, a fabric factory, you know. So that's the main difference. And we're still struggling with some challenges here in Brazil. As an example, I brought you guys here the BST enzyme purification step. We still have some doubts how to perform that, because if you can see here in this SDS gel, the biggest band here, BST 20%, which is a imidazole gradient, we have some impurities, but a lot of enzymes here. And that enzyme actually doesn't work in the reaction. We think it's regarding the purity of that, but we also think about the clarification we are performing. Maybe it's a high temperature, and we have to work on this pureness of this enzyme, because when we have like this BST here on the 40% gradient, it's more pure enzyme, and we are washing this with some amicon, and it's actually working, but you lost a lot of enzymes previously. So we still wanted to learn more about this enzyme and how to improve the production and the quality, because for some basic LAMP reaction, it works properly. But when you go to real samples, we struggle because there is a lot of interferences, especially because they use some fluorophane, like calcium, for example, and it doesn't work. So we're struggling to perform LAMP reaction. We actually have this forecast of producing enzymes for 500,000 exams tests for COVID-19 by using LAMP technology. We are not going to produce the reaction, but we are actually producing the enzyme. So we're still struggling with how to maximize its production, scale up, purity. So we're still learning a lot of things during the production of some enzymes. We started to work with some master mixes, and I got to learn a lot with the Reclone community, but we're still not able to have an ideal master mix here. That's a thing I need to study when I have the proper time for doing that, because right now it's like crazy, and we are working with so many different areas here, such as marketing, quality control, production, lots of troubleshooting, lots of problems to solve, because we are not, besides developing the products, we have to think about how to sell them, supply chain, improving our lab, because it's a private lab, so we didn't start with a huge infrastructure such as at the university. The regulation is such a nightmare here in Brazil, and especially if you wanted to achieve good manufacturing practices and ISO. So we like to joke here that we are not a protein company, we are a paper company here, because there's so many things to fill, many forms, and many things to write. So this is really challenging for a scientist that likes to work at the workbench. You guys might relate with me. We also have this problem of reputation, because as we are national, here in Brazil, we have this problem of, if you are from national production, perhaps we're not good as the German, as the British, the American. So we're trying to turn off these thoughts about us. But hopefully we're gonna achieve this, because our products are will start to be sold in the marketplace here in Brazil. So this is a really step for us, because it brings us a lot of trust, because Merck, Sigma Aldrich, and Merck, if they trust us, so this is a good milestone for us. We don't have, in the beginning, we have the support during the discipline in the university, but as I don't work inside the university, and my supervisor I'm still a PhD student, and my supervisor is not a member of the company, so we don't have all the support from the university, besides some of the professors, the researchers are our clients, but we don't have many support from the university here. So we are really an indie company trying to reach the market and trying to learn everything by ourselves. We never been in acceleration programs or never been doing, we just learning from ourselves and from free sources of information such as the Reclone community and internet so far. And of course, after we have some big sales and proper money to increase the business, we need to increase the staff, the corporative staff, and such as directors in the marketing area and the sales area, and the dedicated quality director, and of course many employees to support this infrastructure. Because as I said, being a factory is a little bit different from being in a laboratory, a research laboratory, and many people is needed to fill those gaps, to ensure that our products are going to be the same every time you produce them, and they have the maximum quality, because it can take our reputation off, you know. And how the community here can help us. Of course, we have to be in touch to learn about every issue we have regarding the production of those components. If you can please share with us protocols, the most optimized protocols for production of those enzymes, recipes for common research lab things such as, for example, protein standard weight, molecular weight. If you could not buy that, even that's very expensive here in Brazil, and it could produce our own and maybe sell those ones, and as an example, would help us to save some money and also to increase our portfolio. It would be very good for us. Example, if you are able to share plasmid with us, with the last high cutting edge, you know, enzymes available, and share with us those types of thing, would also be good to know, to have this, because we need to increase our portfolio, or maybe replace old enzymes with better ones in the portfolio. And of course, sharing our knowledge and troubleshooting. And as Stefan told before, maybe you guys struggle in the past with some issues regarding some products, some enzyme, and we are also struggling with that. So if you know how to solve this problem and you guys can help us, would be great. And of course, the Ecra Biotech and me personally, we also would like to share as much as possible we can, because I think everybody deserves to be independent and don't rely on big players in the market, because we from the South Hemisphere especially, we struggle a lot with that. So that's basically what I have to say about Ecra Biotech. Please visit our website, send me an email. You can guys, you can follow us on Instagram as well. So I hope you guys like the presentation, and hopefully we can be in touch for further explanations regarding Ecra Biotech. So thank you very much.
Thank you so much, Stefan, for that also inspiring talk. I am aware that it is past 4 right now. So I think I could just go ahead and do a brief announcement, and before we go into the question and answer, so that if anyone needs to take off, they are free to do so. Just a basic quick announcement I have, which would come up right now. And if only I can put this in, yeah. Oh. Okay, so my announcement today, so thank you everyone, thank you to the speakers who have spoken to us today. My announcement basically today is to call for volunteers to join us in Reclone and make Reclone fulfill its maximize its potential. So we're calling students to advanced researchers, or maybe you're just a recent graduate thinking about what to do next, wherever you are in the world. We want you. So we have small tasks, we have flexible timing, and of course, we're going to make big impact with this. All that is required is an interest in helping. The areas to volunteer in is to basically make the forum very active and also make the website really functional, and as well as social media. So we have greeters who will help new members feel welcome and keep the forum busy, guides who will show people around and help make connections, recruiters who will help us source for new members and also help with onboarding new members, moderators to keep the conversations on the forum safe and relevant. And of course, there's lots of places to volunteer in terms of communication, organizing meetings, keeping our social media platform active, sharing news, flyers, etc. And of course, we need impact managers to help us know how well we are doing, to let us know whether, of course, what we're doing is making a difference, but we need people who will be able to measure that. How do you get involved? Simple, send me an email at do361@cam.ac.uk and I will be more than happy to let you know how you can get involved. Okay, so that is my big announcement. And I will go ahead and take questions from anyone who may have them right now for either Stefan or Robson. So the floor is open. Please, if you have a question, feel free to unmute yourself and ask. Does anyone want to jump in?
I have one, which is any thoughts of how to come the bias against local manufacturing or the trust issues, because that came up in both Robson and Stefan's conversation. And it sounds like both groups have worked really hard on kind of objective quality in terms of doing the quality controls and making sure it works. But like, how do you overcome that? Because it feels to me like it's not just about data and providing the quality control data, but there's kind of like a broader like cultural habit of maybe buying from overseas. You got any thoughts if there's marketing that could be done or just other suggestions?
Okay, I think I can start. The quality control goes much besides, like having a certificate of analysis of the protein, certificating its activity, its purity, that it worked in your protocol, but go much beyond. It goes like, for example, have a service, a customer relationship. So we have to have an open line to talk with your, let's say, with your customer, but it goes like you have to develop to map like the research that you have been doing to develop that enzyme. So there's certain protocols to entry data to create a new product, but also exit data. I don't know if the translation is right or not, but so we have the research part of the quality control, the production part of the quality control, which regarding you have to ensure that the equipments are calibrated. You have to ensure that your water is good to produce those components. You have to ensure that you have all the qualifications, the certificates from the policy, from the military, because you are using chemical reactants. You have to have a lot of certifications that goes much beyond, just doing the things right. You have to train all the employees in all the security data training. You have to train each equipment. You have to have a training for the employee. You have many feedback, and it's a lot of things, and I still don't know all the issues because it's quite challenging for us to have all those types of quality assurance, because as we are national producers, if, for example, we sell our enzymes for someone that wants to produce a lump reaction, if it doesn't work, the first thing they're gonna call it's us, because it doesn't matter if you produce a good enzyme, but because we have this lack of trust, we are always gonna be the first to be questioned about our enzyme. So you have to ensure that you are delivering a really good product So for that, you have to have many documentation proving that all the process regarding this product, not only the lab process, but everything around this product, you have to have a documentation showing that this is doing the right manner. So this is a lot.
Yeah, I agree. And maybe I'm just wondering about the question about credibility. I wonder if the more researchers use your brand of or like your reagents and actually include that in their publications, so it's known that we used reagents from ECRO Biotech, I think that would give more visibility that we're able to achieve these results with reagents we got from ECRO Biotech. I wonder if that would also contribute to the question about credibility. Just a thought. But any more questions or comments, please, Guy?
Yeah, I have also a comment about this. So it seems to me like, especially right with the COVID now and everything, it makes sense to try to sell this also to sort of the government and as a strategic sort of
that's the right place.
strategic need to have enzymes made in the country locally, sort of, to try to get more support from government, or even if it's the local sort of government. And when I was trying to do molecular biology in Brazil, I mean, it really is hard. Everything costs 3 times more, and if you just sell it for the same price and it comes not in 3 months and not hot, and then it's already a big deal. So, I mean, the universities and the local governments and stuff should see this as sort of a strategic need in general. Yeah.
Stefan, you want to go first, or?
Yes, I can go first. So first, I would like to address, like to add something to what Deborah said. I think it would be very interesting to have some citations of your product being used in particular research article. It's a very nice approach, I will say. But yes, to build the trust, also talking about Jenny's questions earlier, is our approach with, like Robson said, we make sure that our products are of quality for us first, and then we make sure that the customers, once they have the products, they have very good, they provide very good feedbacks, and you do that through constant checking. You have to assist them and really monitor how they feel about the product. So for now, that's the approach we've been up to, and then to maintain our ISO certification, making sure that all the process, the protocols are done in a standardized way. So I think, so about David's questions. So yes, Beneficial Bio is a not-for-profit organization for now. I think that's answering. Okay, sorry.
Oh, well.
Yes, Beneficial Bio is a profit organization. And then, yes, so now we are not expanding to biomass production for now. Yes, thank you.
Regarding Guy question, yeah,
I'm sorry, Robson, yeah, just 1 second, please. Just for the benefit of everyone and for the people who will watch the YouTube video after, now there was a question that was asked, and the first part of the question was directed to Stefan, and that was what he was answering. He said, the question was, I'm impressed on how fast your startup is growing since it's been recently funded. I was wondering, Beneficial Bio is a profit or non-profit organization, which Stefan just answered, saying it's a non-profit organization. And then my other question is for both Stefan and Robson. Recently, we've been thinking and exploring primer production here in Colombia. Do you have any experience with primer production, or do you import them as well? So that would be the question that Robson is going to take a stab at. Okay, go.
Okay. regarding the primer production here, we are not interested in the moment because we have to focus on protein production at the moment. Of course, we have these future plans. Perhaps we can evolve the company for further tools, especially primers is a really interesting one. But we have a national primer production in Brazil here. And they, you know, it's not really easy as just buying a primer synthesis machine here. It's also really important to focus on quality of those products. So because, you know, lots of contamination happens, maybe the synthesis of some primers that are not really good. So you have to ensure that you have a really good quality control regarding purity, regarding the size of the primers production. For that, you have to buy, besides the expensive machine, which is the primer synthesis one, you have to buy a mass spectrometry analysis tool and further equipments to ensure that the primers are really, really good ones. Because in the past, I had bad experience buying some options of primers, and that's a real problem. And you cannot produce at the same place primers and positive controls or protein production, because you always will have contamination in the same site, no matter what you do.
Okay, thank you very much, Robson and Stefan.
Yeah, I just want to echo Robson, because I've been writing a bit on that recently for a handbook we're doing for local manufacturing diagnostics, and the margins on primers are absolutely terrible as well. Like, you would have to sell primers for years and years and years and years to get back your initial investment. The only way that the bigger companies do it is just sheer volume of orders to maintain all of that, and especially with IVD, because they have to have like columns dedicated to certain tests to avoid the contamination and do extremely rigorous cleaning of the columns as well. So my take on that, having looked into it, is it's just not worth it unless you've got a public institution that's willing to subsidize it for the purposes of their researchers having access to. Like, I do know public institutions that have set up primer synthesis machines for research use, and that kind of works, but they're taking a heavy loss. I mean, they're not, it's a very capital intensive setup. So it doesn't, there are some options that are like much more like benchtop now, but they're cheap machines, but they get you on the reagents. So it's still not really cost effective at the moment to do it. But I think the thing is, there's no reason why. I mean, oligos are manufactured very quickly, and they're dry temperature they're ambient temperature shipping. So there's no real reason why they take as long as they do in some places, other than that the distributors seem to be pretty slow at sending them. And there's got to be some level of like supplier distributor shipping arrangements that could get stuff faster to places, and maybe help some of the wait times. But yeah, I wouldn't recommend trying to set up primer synthesis unless you've got a lot of capital and a very long patient time to pay it back again.
Okay, is there any other question? Maybe we have time for one last question, if any, because we have really gone above time, and I thank everyone for staying so far.
There was one in the chat from Natalia.
Oh yeah, thanks. Natalia asks, perhaps going a little deeper in David's question, in Colombia, there is a problem of unemployment of people with PhD, and creation of scientific ventures is proposed as an alternative. However, to make the business viable and perhaps the initial investment looks tall, how have you managed to get those resources to start and maintain the company? So maybe, I don't know, any of you can take a stab on that. Stefan, you're muted.
Yeah, he's muted.
Hi, yes, I was trying to read the question, sorry. So, okay, PhD employment, okay. In our case, maybe I talk a bit, but not in detail during the presentation. We got support from the Schutzwald Foundation and collaboration with the Open Bioeconomy Lab to start that is setting up the local manufacturing unit, purchase of reagents and equipment that we need to start with the local enzyme manufacturing. And the idea is to leverage that as soon as possible from the sales we get and try to be sustainable. So the first step for us will be to be sustainable through our sales and through local sales. And yes, another option is that we are involved in research projects. And it's something also similar to some other research organization here in Cameroon. I take the example of the Center for Infectious Disease, CRID. So they maybe try to address the issue of employment and having maybe postgraduate and researchers involved through research projects. I think since the projects mostly come with the funds, it's just a way of having it. But there's a problem, and then we've been mostly involved with that too. You have to make sure that it doesn't prevent you from doing what you are supposed to do, be it local enzyme manufacturing. So I think it's a double-edged sword. But yeah, our approach was mostly to try as soon as possible to be quite independent and autonomous, sustainable. But for now, we are involved. We still benefit. I think we benefited a lot from the support from the Shuttleworth Foundation, and yeah, we are also involved in research projects. So that's what I can say.
Oh, the Robson wants to say anything about that? You're muted.
Sorry, of course, let's say. Yeah, that's also an issue here in Brazil. In the past, we have lots of investments in science, but the actual government just cutting lots of budget. That's a really a pity. It's a shame. And people are trying to start on the startup after the PhD. We have a lot of challenges here in Brazil, but basically we have some initiatives here to start those government initiatives to support, especially in the state of São Paulo, in which I'm based right now. We have some grants, not so really big grants for life sciences. You know, we need much more money, but some grants to support your business. So we started our company by inviting someone that had a little of, let's say, budget to help us to create basic things. And we are capable to write this grant. We approved this grant here in Brazil. And after that, we actually wrote more 2 grants and won those grants as well. All from the state of São Paulo, but really challenging and sort of exhausting to write the grants, and of course to write the report for those grants. So it's you know, it's good because you can raise money for your company, but you lost a lot of time, especially during the pandemic. For example, I lost a month and a half writing a grant, really huge details regarding everything we would do in this project, and we lost the grant. So we lost many times that we could be able to develop more enzymes here. So yeah, but at least here in Brazil, we still have some things here at the state of São Paulo. So I really recommend to look up for international and national initiatives to support you if you have an idea to create something.
Okay. Thank you so much. I'm just going to read the comment from Jenny and we
close. Oh, don't worry, Deborah, you can wrap up. I was just popping
okay. Okay. Thank you so much to Stefan Fadanka and Robson Tramontina for wonderful presentation today. Thank you everyone for beautiful conversation. Don't forget, this is going to be on YouTube, so you can always go back and have a reflection on it. And also, we're going to post the chats from today on the forum. Please remember, keep the forum alive. You can ask more questions on the forum and just engage. So thank you until I would say this in a funny way, until we come your way next time with another edition. Have a lovely rest of your week. Okay, bye.
Ciao, ciao.
Thanks. Bye.
Ciao.